I know some of you are continuing to get your lunches, but I'm going to begin the announcements. Thanks for coming today to Grand Rounds, the Department of Psychiatry. And I have the pleasure of introducing Sunila Nair, who will be giving our presentation today. Sunila has been an assistant professor in the department since 2015, and her training background was that she graduated with a Bachelor of Medicine and Surgery, an MBBS, in 1998 from the University of Mumbai. And then she came to the US and did her PhD with Gary Gudelsky at University of Cincinnati, where she used did some neurotoxicity and some behavior and neurochemistry, looking at MDMA and cholinergic neurons. And at that point, she moved to more behavioral models and worked with Yavin Shaham at the National Institute on Drug Abuse for several years, for 4 years. And she, at that point, decided to move to Seattle and came into our department and worked with me and with some of my colleagues and set up some very sophisticated behavioral models that I think you'll be hearing some more about her strategies today. But when she became an assistant professor, she quickly moved through some successful grant applications. And I think that any major grant that she's written, she's received. So she's got a perfect record in my. And I think that reflects her brilliance and her scholarship. And today she's going to tell us about some of the work she's been doing over the last several years and also some of the directions for her future research. So please join me in welcoming Dr. Sunila Nair.
Thanks, John, for the really kind introduction. I hope my talk lives up to that, and thank you all for coming, and thanks to the department for inviting me today to do Grand Rounds. So we're a preclinical basic science research lab, and I'm going to talk to you today about the behavioral and neurochemical mechanisms of relapse to cocaine seeking in a rat model. So here's a brief outline of my talk. I'm going to first talk about the clinical problem, the huge clinical problem of addiction. I'll then introduce to you the lateral habenula, which is a really tiny brain region that we have been interested in for the last few years in understanding its role in addiction. I will then talk about some tools that we use in the lab to modulate the activity of lateral habenula neurons, virus-mediated gene transfer, and DREADS. And I'll then show you data on modulating the activity of lateral habenula neurons, what it does to ongoing cocaine and food self-administration, as well as relapse to cocaine-seeking behaviors. And then finally, I'll talk about more recent data where we have started looking at sex differences in addiction. So here's a brief background about cocaine. Cocaine is actually produced naturally in the leaves of the cocoa plant, which is a shrub that is indigenous to the foothills of the Andes in Peru and Bolivia. So the cocoa plant was basically reserved for royalty among the South American Incas, but it was soon found that the native people can be made to work harder and longer in the rarefied atmosphere of the Andes if they were allowed to chew these cocoa leaves. So typically what the user did was made a ball-like quid from these cocoa leaves along with some alkaline material like lime. The quid was then held in the cheek where saliva leached the cocaine from the leaves, and this produced mild euphoria, suppression of hunger, and increased physical stamina. Over the next couple of centuries, sporadic attempts were made to bring these magical leaves to Europe, but more often than not, the leaves did not survive the journey. And it wasn't until 1863 that Italian chemist Angelo Mariani, having heard of these cocoa leaves, he devised a fortified wine that he called Vin Mariani, which he made by mixing these cocoa leaves with Bordeaux wine. The wine became immensely popular in the 19th century. Queen Victoria tried it. It was even endorsed by the Pope, who gave it a papal medal. In the United States, John Pemberton, a pharmacist in Atlanta, produced Pemberton's French wine cocoa, which is basically Vin Mariani, but he flavored it with cola nut extract, and that contributed caffeine to the cocktail. When Prohibition set in, he devised a new drink where he replaced the alcohol with sugar, and that's what we know as Coca-Cola. So this is interesting, but we know that cocaine addiction is a very serious health issue. And just to illustrate how serious the problem is, 1 in 4 Americans between the ages of 26 and 34 have used cocaine at least once in their lifetime. 5,000 adults try cocaine for the first time every single day. Nearly half of all drug-related ER visits are due to cocaine abuse. And in 2009, 300,000 infants were born addicted to cocaine. And this is really interesting. Cocaine binds really closely to the ink and paper currency. FBI chemists have discovered that traces of cocaine can be found pretty much on every dollar bill that is in circulation. So I hope you'll appreciate that addiction. It's a very debilitating and really costly neuropsychiatric disease. And the disease itself is characterized by uncontrollable drug craving, compulsive taking of the drug and seeking for the drug, and a very high propensity for relapse. About 40 to 60% of patients who have been addicted to drugs will relapse to the same drug or to another drug. And despite this, only about 10% of patients are in treatment, and this is in part due to the stigma that is associated with the disease. So now I'm going to switch gears a little bit and introduce the lateral habenula, which, like I said, is the brain region that we have been interested in studying for the past few years to understand how it fits into the neurocircuitry of addiction. And what you're looking at here is a sagittal section of the human brain. And the habenula is this tiny nucleus right here next to the thalamus under the hippocampus. In humans, deep brain stimulation of the lateral habenula has been used for the treatment of refractory depression, although the mechanism that really underlies the therapeutic effect of DBS in alleviating depression is not understood. This is a sagittal section of the rodent lateral habenula, and it's a schematic, and you can see that the habenula is divided into medial and lateral subdivisions. The lateral habenula itself, again, has numerous different nuclei. It's a very heterogeneous brain region, but for the purposes of today's talk, we're not going to focus on different nuclei of the lateral habenula, but rather treat the nucleus as one single entity. The lateral habenula has like a really wide range of physiological functions as well as pathological, including the regulation of circadian rhythms, maternal behavior, regulation of stress responses, nociception, learning, and reward. And reward is the behavior that we are most interested in. The lateral habenula, if you just Google lateral habenula, you'll see that the word that comes up often is anti-reward. So the lateral habenula is known as the anti-reward nucleus. And to explain what that is, I'm going to first show you what reward looks like in the brain. So this is a very classic study by Wolfram Scholz looking at the firing of dopamine neurons in response to a reward. And the reward here was it was a sip of fruit juice. So when an animal is presented with a rewarding stimulus, dopamine neurons fire. Then what they did was they paired this rewarding stimulus with a conditioned stimulus, which predicted the reward. And when they exposed the rats to the conditioned stimulus, the dopamine neurons fired in response to the conditioned stimulus. And when the reward was presented a few seconds after the conditioned stimulus, there was no change in activity in these dopamine neurons. And then finally, when the conditioned stimulus was again presented, the dopamine neurons fired. But when the animal was expecting a reward but did not receive it, the dopamine neurons stopped firing. So there's decrease in firing of dopamine neurons in the absence of reward. In contrast, if you look at the firing activity in lateral habenula neurons, the red circles are in the presence of reward. You will see that the lateral habenula neurons decrease their firing in the presence of reward. In contrast, when there is no reward, there is an increase in firing in lateral habenula neurons. So this is exactly the opposite of dopamine neurons, which fire in the presence of a reward and do not fire when there is no reward. And this is a raster plot, similar to what I showed you in the previous slide. In the presence of reward, lateral habenula neurons stop firing. And if you look at this graphic here, um, this is showing you data from the lateral habenula, which is showing you a decrease in firing, and again recording from the mediodorsal thalamus, which is the nucleus right next to the lateral habenula, and that doesn't do anything. So this is an effect which is very specific to the lateral habenula, and it's the anti-reward center of the brain. So we were interested in understanding if the lateral habenula influences cocaine taking and seeking behaviors, and in understanding what is the underlying neuronal circuitry, what is the molecular signature of lateral habenula neurons that is activated either by drugs or non-drug reinforcers, and does the lateral habenula have any role to play at all in disease progression from just casual cocaine use to cocaine addiction? So in the lab, there are several animal models that are used to study addiction, and this is by no means a complete list, but I just want you to appreciate that addiction is a very complex disease, and it really is a continuum from like single or casual use of the drug to um, escalation in use to habit formation and eventually abstinence and relapse. And each of these models and each of these processes recruits different brain regions, and each of these models really targets a very different phase of addiction. And the models that I will be talking about today that we use in the lab are intravenous self-administration, extinction, and reinstatement. And I'll talk a little bit about some data, recent data that we have on incubation of craving. So for our experiments, so this is in a rat model. We make silastic catheters that we implant into the jugular vein of the animals, and after a post-op recovery period of about 7 to 10 days, the animal is put in an operant box. So what you're looking at here is the animal pressing a lever, which is called the active lever. And when the animal makes a lever press on the active lever, he gets a single injection of cocaine through this infusion syringe connected to the infusion pump, and along with it, signaling cocaine is a cue light. And what you can't see here is that the animal also gets a tone that signals cocaine. So when the animal presses the active lever, he gets cocaine and he gets cues that are associated with cocaine. And I'm not sure you can see here, but there is a second lever on the back end of the box, which is the inactive lever. And if the animal makes a lever press on this lever, he doesn't get anything. So there are no programmed consequences to making lever presses on the inactive lever. And this is what a typical experiment looks like. On the y-axis is the number of lever presses. On the x-axis is experimental days. The first phase of the experiment is self-administration training, which, like I said, is the rat is put in the operant box and he's trained to self-administer cocaine. And this happens over a period of 10 to 15 days. The animal lever presses, increases responding over the first few days while he's trying to learn to self-administer cocaine, and eventually the responding stabilizes. And we think of this phase as mimicking casual drug use. So it's a very simple paradigm where the animal makes one lever press and gets a single infusion of cocaine. It's called a fixed ratio, one schedule, and we think of it as mimicking casual drug use. In the second phase of the experiment, which is called extinction, and this is in a drug-free condition, drug is not available, the syringe is taken away and the animal lever presses. When the animal lever presses, he doesn't get anything. So the animal quickly learns that the lever presses are not associated with cocaine, and responding comes back down to baseline. And in the third phase of the experiment, which is really the relapse test, which we call reinstatement in the lab, the animal is exposed to either a single priming injection of cocaine, or it's exposed to cues, the tone and light cue that I said was associated with cocaine delivery, or is exposed to a stressful stimulus like a foot shock. And all of these conditions, which are known to induce relapse to drug seeking in humans, will also cause reinstatement of lever responding in our rat model. So these are data from a self-administration experiment. On the y-axis is the number of infusions or lever presses, and on the x-axis is the cross-training days. In green is the number of inactive lever presses, so the animals learn very quickly that the inactive lever doesn't give them anything. In the red is the number of infusions that the animals self-administer. And in blue is the number of active presses, that is the total number of presses that the animal makes even when cocaine is not available. And these are extinction data. So when the cocaine syringe is removed and cocaine is taken away from them, the active level responding, eventually it first starts off high and then eventually it comes back down to baseline. And these are reinstatement data when the animal is exposed to a single injection of cocaine or is exposed to cues or exposed to stress. In this case, it was a foot shock. All of these conditions produce reinstatement of level responding in the animal model. So everything I talked to you was about drugs of abuse, specifically about cocaine, but interestingly, exposure to stress or access to palatable food or cues that are associated with food, it can also produce relapse to unhealthy eating habits. And this is really seen when people are dieting. And to study this, when I was a postdoc in Yavin Shaham's lab, I adapted the reinstatement model, which is really very commonly used to study relapse to drug seeking, to study relapse to food seeking behaviors. The model is really very simple. The rats are trained to self-administer food instead of drugs, and then the food is taken away from them during extinction, and then exposure to a non-contingent pellet, that is one single pellet, just the taste and smell of the pellet, or cues or stress. All of these conditions produce reinstatement of level responding to food. So in the lab, to manipulate the activity of lateral habenula neurons, we use chemogenetics. And I'm going to talk to you a little bit about what it is, because some of you might not be familiar with it. And it's really based on a very simple concept that neurons signal via receptors. And what you're looking at here is a pre- and postsynaptic terminal. Neurotransmitters are released and they act on receptors in the postsynaptic terminal, and the activation of these receptors stimulates various signaling cascades. So for chemogenetics, what is done is that these receptors are mutated, and instead of having affinity to its native ligand, it now has affinity to a ligand that is otherwise inert. And the chemogenetic tool that we use in the lab is called DREAD, which stands for Designer Receptor Exclusively Activated by Designer Drugs. And like I said, these receptors are mutated receptors. In this case, they're mutated muscarinic receptors that have lost their affinity for acetylcholine, which is the native ligand. And instead, they now have high affinity for CNO, or clozapine N oxide, which is otherwise a mostly inert drug. So dreads come in many flavors. So like I said, these are artificial receptors, and depending upon what signaling cascade they stimulate, there are GI-GO coupled dreads, or GQ coupled dreads, or GS coupled dreads, and also arrestin dreads. And depending on what signaling dread you're using, they either activate, or very simplistically, they either activate a neuron or silence the activity of the neuron. But all dreads, activation of all dreads will transiently alter cellular activity by increasing G protein coupled signaling cascades. So now we have this artificial receptor that we want to inject into the lateral habenula to see what it does to addiction behaviors. But there has to be a way to get this receptor into the neuron. And we use viruses for it. And the virus that we're using for these experiments is adeno-associated virus. And what you're looking at here is lateral habenula neurons that are infected with AAV. So before we started doing experiments, like I said, dreads come in different flavors. Similarly, these viruses, there are several different serotypes of the viruses, and each of them perform slightly differently. So we tested 2 different serotypes of the AAV virus, 5 and 8, and you can see it's pretty obvious that the 8 serotype really produces very robust infection compared to the 5 serotype. The first experiment we did was to validate if this procedure actually works or actually does what we think it does under our experimental conditions. And to do that, we tested the effect of CNO on CFOS, which is an immediate early gene that gets activated in response to neuronal stimulation. And we used cocaine here, a single intraperitoneal injection of cocaine to activate CFOS. And in the presence of the GI-GO coupled dread, you can see there is a significant decrease in CFOS cells in the lateral habenula. And with the GQ dread, there is a significant increase in CFOS. So this gave us confidence in our techniques, and we went ahead to do our behavioral experiments to try and understand what activation of G protein coupled signaling does to first the self-administration phase of the addiction cycle, and then the reinstatement phase. So these are experiments that were done by Dennis Smirnov, who was a first an undergraduate student and then a research tech in the lab. He's now an MD PhD student at UCSD. And Melissa Estabrook, who was an undergraduate student in the lab and now is in medical school here at UW. So what Dennis and Melissa did was injected these animals with a GI-GO coupled dread that silences lateral habenula neurons and trained the animals to self-administer cocaine, and then injected the animals with either 1 or 3 milligrams per kilogram of CNO. And in the blue bars are the number of infusions that the animal self-administers, and the red is the number of active levopresses. So as you can see, there is some increase in both the number of infusions and active levopresses, but this is not statistically significant. In contrast, at 3 milligrams per kilogram of CNO, there is a significant increase in both the number of infusions as well as the number of active presses that the animal makes in the 2-hour period. And at the bottom are time course data. So these are 2-hour sessions, and this is the time course across those 2 hours. And you will see that the most significant effect is in the first 30 minutes of the test session, and that is when the animals are loading the cocaine. So what these data mean is that when lateral habenula neurons are inhibited with CNO, the animal finds the dose of cocaine that the animal has is not as rewarding, which is why the animal has to levopress more to maintain brain levels of cocaine. So whenever we do these kind of experiments, it's really important to rule out alternate interpretations of the data. And one thing that could have happened here is that in the presence of CNO, the animals could have just moved more. There could be just an increase in locomotor activity. So what Dennis did was he took animals and put them in a locomotor activity chamber, which is basically a small box here which has beams across it. So when the animal moves, it breaks beams, and that's what we record and use it as a measure of locomotor activity. And Dennis found that CNO had no effect on locomotor activity at the doses of CNO that we used in our experiments. These are data from Sharif Taha's lab, and they did similar experiments with alcohol instead of cocaine, and they also used slightly different techniques. This is not as nuanced as using dreads. What they did was they electrically ablated the lateral habenula, but the results are really parallel to what we found with cocaine. So this is from a 2-bottle choice paradigm. And the closed circles are animals that were lesioned, and the open circles are animals that were sham lesion. And you can see very clearly that the animals with the open lesion, there is an increase in alcohol intake. They found similar results using operant techniques like alcohol self-administration, just like we use in the lab. There is an increase in the number of levopresses, increase in ethanol intake, as well as an increase in blood ethanol concentration. So this is very interesting because it shows that it gives us more confidence in our results, and it suggests that inhibiting the lateral habenula, it really decreases the reinforcing value of the drug and increases levopressing so that the animals can maintain their blood levels for the drug. We then tested what inhibition of the lateral habenula does to food self-administration. And this is, again, a very simple experiment where animals are trained to self-administer food, and once the responding is stabilized, they get either vehicle or CNO. And as you can see, there is no significant effect of inhibiting the lateral habenula on food self-administration. And this is not very surprising because, like I said, the lateral habenula is activated in response to aversive stimuli. And just simple ongoing food intake, there's really not much aversiveness associated with ongoing food intake. So this is an interesting control experiment for our cocaine results. So I showed you data from this phase of the addiction process, we then looked at what inhibiting the habenula does to reinstatement of cocaine seeking. And we did this in the presence of a cocaine prime. So when the animals were extinguished, we gave them a single priming injection of cocaine and looked at what that did to reinstatement of cocaine seeking. And what we found, so on the y-axis is the number of active presses. This is with an injection of just vehicle, and in the presence of CNO, there is a significant decrease in the number of active presses. And again, this effect is most predominant in the first 30 minutes of the test session. We saw a similar pattern when the animals were exposed to contingent cues to reinstate lever responding. So this is the number of active presses in response to vehicle. And again, CNO produced a significant decrease in the number of active presses. So what this suggests is that during reinstatement conditions, when cocaine is not available, the lateral habenula neurons are active because of the absence of cocaine, and inhibiting the lateral habenula neurons decreases reinstatement of cocaine seeking. So we then looked at what this effect does to reinstatement of food seeking. And these are data from food self-administration. So the animals learn to self-administer food. This is the number. This is inactive level pressing. This is the number of pellets that the animal self-administered, and this is the number of active level presses. During extinction, just like with drugs, level responding comes back to baseline. And when the animal is exposed to a single pellet, just the smell and taste of the pellet reinstates level responding, but there is no difference when the animals are injected with CNO. So basically, inhibition of the habenula, it decreases reinstatement of cocaine seeking, but has no effect on reinstatement of food. We then did a couple of experiments in the lab to really rule out the possibility that CNO, which is supposedly an inert ligand, but we wanted to rule out the possibility that CNO had any effect by itself. And these experiments were done by Ashley Evans, who's here today, and Philip Silva. So what Ashley and Philip did was they did one experiment where no viral vectors were injected into the lateral habenula. So these animals were only implanted with jugular catheters, trained to self-administer cocaine, and we looked at the effect of CNO on cocaine self-administration and reinstatement. And then they did another cohort of animals where they injected the animal with only the reporter gene, EGFP, without the DREAD transgene, and looked at what that did to self-administration and reinstatement. So as you can see, with no viral vectors in habenula neurons, there is no effect of CNO either on cocaine self-administration, on priming reinstatement, or on Q reinstatement. Similarly, in rats that were injected with GFP in habenula neurons, there is no effect on any of these behaviors. So this suggests that really gives us more confidence in our results and suggests that CNO really doesn't have any off-target effects under our experimental conditions. So everything that I've shown you so far is from inhibiting the habenula using a GI geocoupled DREAD. So the beauty of using DREADs is that you can really use the same technique to both inhibit as well as activate the neurons using the exact same technique. So we asked what activation of the lateral habenula using a GQ coupled DREAD does to these addictive behaviors. And in our first experiment, we injected the GQ-coupled DREAD in the habenula and looked at what it did to cocaine self-administration as well as food self-administration. And like you can see, it produced a very robust decrease in both cocaine intake as well as pellet intake. We then did our control experiment to see if this effect was due to any kind of deficit in locomotor activity, and indeed it looks like that was the case here. So again, this is in a locomotor activity box, and we did like several doses of CNO. The doses of CNO that we used in our experiment produced a significant decrease in locomotor activity, suggesting that the results that I just showed you in the last slide were at least partly due to some deficit in the motor activity in these animals. We also did another test that looks at motor behavior, and this test is called the rotor-rod test. This was done by Melissa Estabrook, and it's really a very simple behavioral test where the animal is put on a rotating rod, and the amount of time that it takes the animal to fall off the rod is measured. So what you're looking at on the y-axis is the rotor-rod score, which is really the number of seconds that it takes the animal to fall off the rod. And with both these doses of CNO, there was no effect in the rotor-rod test. So the same doses of CNO that produced a significant decrease in locomotor activity had no effect on the rotor-rod. So this suggests that the effect of activation of GQ-coupled signaling in lateral habenula neurons is really very complex and nuanced, and it's really to our advantage to pick doses of CNO before we start these experiments that do not have any effect on locomotor activity to see what it does to see that the effects are exclusively due to modulating GQ coupled signaling. So I hope I've convinced you that the lateral habenula is neurons in the lateral habenula involved in both casual cocaine taking as well as seeking behaviors. The next step for us was to understand what is the neuronal circuitry that underlies these behavioral effects. The lateral habenula projects to several different brain regions. And for our experiments, we targeted two specific brain regions, the ventral tegmental area and the VTA, and the rostromedial tegmental nucleus, the RMTG, because both of these nuclei are known to be involved in addictive behaviors. The lateral habenula also projects to the raphe. I should say that before John Newmeyer has a panic. But we didn't study the raphe projection in our experiments here. The technique we use to target projection specifically from the lateral habenula to the VT or RMTG is by using dual intersectional genetics or dual vectors. So the first vector that we used is a doubly floxed inverted vector, a DREAD vector, which was injected into lateral habenula neurons. And the second virus that we used was a canine adenovirus that expresses pre-recombinase. So the way this works is that the canine adenovirus gets transported retrogradely. So when TREAD, the inverted transgene, is injected into the lateral habenula, and canine adenovirus expressing pre-recombinase is injected into the target region, it gets taken up by these neurons in the target. It gets transported to the lateral habenula, where it flips the inverted transgene into its proper orientation to allow for transcription to occur. So using this technique, we targeted lateral habenula projections exclusively to either the VTA or the RMTG. And these are data from inhibiting this projection from the VTA to RMTG on cocaine self-administration. And as you can see, we found no effect on either the number of infusions that the animal self-administered or the number of active lever presses. Similarly, when we did this experiment targeting the lateral habenula projections to the rostromedial tegmental nucleus, there was a slight increase in the number of infusions and lever presses, but this was not statistically significant. We then looked at reinstatement of cocaine seeking in response to a priming injection of cocaine. And this experiment was done by Philip Silva when he started out in the lab. So what Philip did was injected both the viruses into the lateral habenula, implanted catheters, trained animals to self-administer cocaine, extinguished their behavior, and then injected them with a single injection of cocaine with either vehicle or CNO. And though there was always a partial decrease in the number of lever presses, this was never statistically significant. So basically, I guess we could conclude that this pathway is not involved in either self-administration or cocaine priming reinstatement. But I think we need to take a step back and fully appreciate that this technique that we have used may not have targeted the adequate number of neurons that are required to manifest the behavior. So what Philip is doing now is that he's injecting only one virus, dreads, into the lateral habenula, and instead of injecting CNO systemically into the peritoneal cavity, he's injecting CNO directly into the ventral tegmental area or the RMTG to try and get more neurons activated so that we can completely either rule out or see if these pathways are involved in this behavior. So to conclude this part of the talk, activation of GI-GO-coupled signaling, it enhances operant cocaine self-administration, which models casual drug intake. And it also decreases reinstatement of cocaine seeking, which basically models cocaine addiction. And I think these data are really important from a therapeutic perspective because it's really important to know that this same manipulation that has one effect on one phase of cocaine self-administration has an entirely different effect on a different phase of cocaine self-administration. So one therapy that may be effective for one person may not be effective for somebody else because they're just in a different phase in the addiction process. And also, activation of GI-GO-coupled signaling had a differential effect on reinforcement induced by drugs versus non-drug reinforcers. So to summarize, the lateral habenula is an important component of neural circuitries that underlie both recreational cocaine intake as well as cocaine addiction. I'm now going to talk about a newer project that we have started in the lab that's led by Ashley Evans, and looking at sex differences in addiction. So a little bit of background here. Although males are known to have greater access to drugs of abuse in general, escalation of drug use is significantly greater in females compared to males. The transition from drug use, just casual drug use, to addiction is more rapid in females. When women seek treatment, they're consuming significantly greater amounts of drug compared to males, and females also report greater craving and relapse behaviors. The behavior that we were interested in studying was incubation of craving. And what incubation is really a fancy term for a time-dependent increase in craving during withdrawal. And these are two studies from in humans looking at incubation of cocaine craving and incubation of cigarette craving. So what you're looking at on the y-axis is craving score, and this is an inpatient clinic, and you can see the craving is decreasing, and this reflects the day after discharge, craving ratings after discharge, and there is a time-dependent increase in craving both in individuals that relapsed as well as in individuals who are still abstinent. And a similar pattern is seen with incubation of cigarette craving when people were exposed to either neutral cues or a smoking cue. In the lab, the way we model incubation of craving is that we train animals to self-administer cocaine for about like 10 or 14 days and bring the animal back into the self-administration context, either one day on day one of withdrawal or at various different time points after withdrawal, going up to many months. And these are representative data. So this is the number of lever presses and the number of withdrawal days. And as you can see, there is a time-dependent increase in the number of lever presses across withdrawal days. However, this doesn't go on infinitely. And it comes back down. This is for cocaine. These data are for cocaine. And approximately in 180 days, you see that craving starts to come back down. So to look at sex differences in incubation of craving, what Ashley did was that she trained both male and female rats to self-administer cocaine for 10 to 14 days and then brought the animals back into the self-administration context on day one. And on day one, there is really not a significant difference between males and females. But when she did it on day 35, there is a significant increase in lever pressing in males, and so you see incubation of cravings, but you found this massive increase in lever pressing in female rats, And interestingly, this effect is only seen in female rats that are in the estrus phase of the hormonal cycle, and not in female rats that are in non-estrus phases of the hormonal cycle. So estrus phase is when when estrogen levels are relatively high compared to the other three phases when estrogen levels are relatively low. So what we're doing now is working under the hypothesis that this potentiation of level responding is mediated by estrogen and estrogen receptors. And there are two estrogen receptors. They're both nuclear receptors that we're looking at, alpha and beta. And in initial experiments, we are characterizing anatomically estrogen receptor expression in lateral habenula neurons and asking the question if there are any cyclical hormone-dependent changes in estrogen receptor plasticity in these neurons. And this is done, Ashley and Sarah Claypool, who's an undergraduate student in the lab, are leading this. So what Ashley and Sarah are doing are taking rats, females, so this is in female rats, and taking vaginal swabs from these rats and looking at it under the microscope and classifying these animals into the 4 phases of the estrous cycle that we see in rodents. And then we take the brains from these animals and look at the expression of estrogen receptors alpha and beta in lateral habenula neurons. And I'm not sure if you can see that. I think you can see this, but the green is the number of estrogen receptor alpha, and the red is beta. And we are looking at the distribution of these receptors across the rostrocaudal axis of the lateral habenula in the estrus phase as well as the non-estrus phases. And what we find when we look at the total number of estrogen receptors is that there is a decrease in the total number of receptors, both alpha and beta receptors in the estrus phase of the estrous cycle compared to the non-estrus phase. And when we count the total number of cells that contain estrogen receptors, they're always lower than the total number of receptors, which suggests that there are multiple receptors that are localized in a single cell. But it follows the similar pattern that in estrus there is a decrease in the number of alpha as well as beta cells. This is more clearly depicted in the pie chart. So if you look at this non-estrus pie chart in non-estrus, about 1/4 of the receptors in the lateral habenula express estrogen receptors in non-estrus phases, of which there is almost an equal distribution of alpha beta receptors as well as cells that contain both. And there's a significant decrease in estrus. And we also looked at the gradient distribution of receptors from the anterior to the posterior habenula. And what we found was that in the anterior habenula, there is a greater distribution of expression of the alpha mRNA compared to the beta. And in contrast, in the posterior habenula, there is a greater distribution of the beta mRNA compared to the alpha mRNA. So just to summarize these data, estrogen receptors, both of the alpha and beta subtype, they're distributed across the rostrocaudal axis of the habenula. And there is a gradient to this distribution. The alphas are primarily localized in the more anterior parts of the lateral habenula, while the betas are more predominantly expressed in the posterior habenula. Both expression of both alpha and beta is estrous cycle dependent, and the expression of both receptors decrease in the estrous phase of the estrous cycle. And the expression of both these receptors in individual neurons is also decreased in the estrous phase of the cycle. So going forward, what we're doing is trying to knock down these receptors in the lateral habenula using CRISPR-Cas9 technology to see if estrogen and estrogen receptors actually have any influence on cocaine taking and seeking behaviors. And I think I'm going to stop there so that there is enough time for questions. I'd like to thank everybody in the lab who I think I acknowledged along the way for all of their hard work in getting all these data. And I'd like to thank John Newmeyer for being a great mentor. Everybody in the Newmeyer lab, I really feel grateful that I get to work with them on a daily basis. Robert Steiner in the Department of Obstetrics and Gynecology, who has been guiding me in our new project on sex differences and helping me with understanding the really interesting physiology of female sex hormones. Adam Blashak and Kevin Coffey, I didn't show you this data today, but we work with them on identifying the molecular signature of neurons in the lateral habenula, and Kevin is helping us with recording ultrasonic vocalizations to determine the affective state of the animal during self-administration. And Scotting Evans, who is really helps us with all of our behavioral equipment and magically keeps everything going. And I'd like to thank my funding sources, ADAI here at the UW, the Brain and Behavior Research Foundation, and of course, NIDA. All right.
Are there any anecdotal or like natural experiments that show a difference in behavior in patients who rest had a lesion in the lateral habenula? Are there any are there any examples of like someone who had a stroke in that particular area and then
Interesting. Yeah, that's a good question. So the question is, are there any examples of patients who have had strokes in the lateral habenula region where there have been differences in addiction behaviors? And I'd have to say I'm not aware of any such studies. The lateral habenula, so in humans, I don't think I mentioned this, but the habenula is, so we're targeting the lateral habenula. In humans, it's very hard to differentiate the medial and lateral. It's a very small nucleus, and fMRI studies and other PET studies, they really treat it as a single nucleus. You can't really differentiate between the medial and lateral. And what we know from animal studies is that the medial and lateral habenula are really very different with respect to neurons that project to it, the neurons that efferents that project out of it, as well as the molecular signature of neurons in these two brain regions. So yeah, that's a good question, but I don't know the answer to it. Yes.
I'm just wondering about the difference between food and cocaine and what you found. And I'm just wondering if you did something like food deprivation and were found as strain of mice that overate, do you think the response to the lateral habenula will be different? And does anyone look at that sort of thing?
Yes, that's a great question. And no, nobody has looked at it, but that's something that I really want to study. It's that, as well as like models like binging or anorexia, where there's like a negative affect associated with it. I think the lateral habenula will be part of the circuit under those conditions.
And if you could just, I know this is kind of a tough question, but I always kind of think about regions in the brain as parts of systems. And I struggle with habenula, like what system is it part of? Like what would you characterize? Is it part of, what would you say here?
Well, I would say it's part of the reward circuitry in some ways. It's like anti-reward. It also has like many other behaviors. So this is a very microscopic view because this is what we study, but there are lots of studies looking at habenula in maternal behaviors as well as regulation of stress responses. So I wouldn't say so, though I think of it as part of the reward circuitry. That is really not the whole picture. And it's basic. A lot of people used to look at it before the Nature study came out in 2007. The habenula was mostly of interest to anatomists who looked at it as like a relay station, looking at inputs and outputs. And it was only after that study came out that people who study addiction thought that, you know, you should pay more attention to that nucleus. And yeah, that's how we started doing our work.
Right.
Yeah.
Because the follow-up question would have been, what kind of process do you think might be mediated or facilitated by habenula? Like what sort of decisional process? Is it setting the reward expectancies, or is it discrepancies, or?
Yeah, that's a very good question, whether the habenula is just a relay center or if it actually processes information to decrease the value of the reward or not. And I don't know the answer to that. The studies are just, we're ongoing. Yeah.
The party line is probably not correct, but the party line is that those are mostly glutamatergic neurons that project mostly to GABAergic neurons in the other areas, and they can put the brakes on dopamine, put the brakes on serotonin, but it's clearly, that's not the full story.
And one more question. So
Yes.
I've really got into sleep recently and dopamine release of sleep and how it's involved in memory and behavior. And I'm wondering if you've ever looked at how sleep affects this system as far as the reinstatement and or, you know, have you ever thought about doing that? Is there any evidence in the, because I know you do this stuff for multiple days. I don't know if you,
Right. So we do it for multiple days and we do it in the same circadian cycle every day. And we haven't switched circadian cycles to look at what like sleep would do to, but that's a very good, very interesting idea.
All right. Thank
Great.
you.
Thank you.
Cheers.